when we talk about the whole point of mitosis. Okay, mitosis is used in all cells in an organism with the exception of sex cells. Sex cells use meiosis. I didn't name these, not my fault, they're similar, but mitosis is used, it's an asexual form of cellular division. Asexual means it makes identical copies. But the whole point of mitosis is to actually ensure that the DNA in a cell is evenly divided. Okay? The organelles, so enough mitochondria will end up in each daughter cell, enough endoplasmic reticulum, and all that other stuff. Okay, all those other organelles, the cytoplasm will divide roughly equally, but it's the DNA that's super important, because if DNA does not divide unbelievably equally, then those two daughter cells are not going to survive. They're going to be missing a chunk of important DNA. So mitosis's whole point is to ensure unbelievably equal division of DNA material. When we start off with mitosis, we're starting off with a regular cell, okay? And that regular cell has what we call chromatin material. So its DNA inside its nucleus just looks like everyday DNA. Now I'm going to draw this to just look pretty darn regular here. So it's got a bunch of DNA, got more DNA and more DNA. So that's the DNA in the cell. That cell is just doing what that cell's supposed to be doing. And when a cell is busy being that cell, whether it's a muscle cell, an eyeball cell, or whatever, we say that that cell is in a stage of its life called interphase. An interphase is where the cell is doing what that cell was made to do. So it's, retracting, contracting, expanding. If it's a muscle cell, it's, absorbing light. If it's in the retina of the eyeball, whatever. Okay? When that cell gets the message, okay, that it has to divide because we need more cells, it starts the process of mitosis. So the first stage of mitosis sees the actual nucleus here getting more and more invisible. It's starting to break apart because we know that this nuclear membrane right here, okay, is actually made up of parts of the cell. It contains all of the DNA. And in order for DNA to actually divide, we have to get rid of the nucleus. So all the organelles in the cell push off to the extremities, so surrounding the cell membrane, so that we're left for tons of space for the DNA to properly divide. Okay? So from there, when we sense the beginning of cellular division, the cell itself stays the same size, but we notice that the nucleus is starting to break apart. Okay? So there's our nucleus. The second thing we notice at the beginning of cellular division is that we go from having a bunch of mixed up DNA to distinct chromosomes. So you'll notice that in prophase, okay, the nucleus is starting to disintegrate, and we start to have chromosomal formation. Okay, that's where my little X's come up. And we've got these two organelles. These two little organelles are called centrioles. Don't worry about that much. Just know that you get one from your egg and you get one from the sperm cell. So when a sperm cell combines with an egg, it actually gives the egg two things. It gives the egg 23 chromatids, and it gives the egg a centriole. Because we need, for every cell that divides, we need two centrioles inside it. They're the things that are going to make those clotheslines, right? Going from one end of the cell to the other for the chromosomes to actually hook up to, right? Because we remember, deep down, we remember that a chromosome has one sister chromatid, another sister chromatid, and it's got that centromere in the middle, right? And it's the centromere that's going to hook up to stuff. It's going to hook up to that clothesline. So if we want cells to divide right now, we can't really just pull them apart as they are. This would be messy and it would not be even. Okay, we would have, these two cells going this way and those two cells going that way. Disaster. We don't want that. For mitosis, the beauty of mitosis is that it ensures 100% equal division of DNA. So every one of these actual chromosomes here, okay, this side right here is identical to this side right here. These two sister chromatids are identical to one another. So we got to make sure if they're going to divide these chromosomes that 1/2 of each chromosome goes into each new cell. Okay? So in order to do that, they all line up in the middle of the cell. Okay, the halfway point of cellular division is known as metaphase. And by the time metaphase rolls around, the actual DNA, the nucleus is completely gone. And what ends up happening out of these centrioles is we've got these clotheslines. Okay, they're actually called spindle fibers, and they go from one end of the cell to the other. All the organelles are off to the side. We're not even drawing them. And on every one of these clotheslines, one of the chromosomes is going to hook up. Okay? So one of these chromosomes hooks up to every one of these clotheslines, these spindle fibers. So we remember that if this is a human cell, this is going to actually have 46 clotheslines. Okay, there are going to be 46 chromosomes, one in front of the other, and it doesn't matter how they line up. You could get chromosome 1, then chromosome, 23, and then 18, and then it doesn't matter. Okay? They all line up through metaphase. And then the magic starts to happen. Then we actually see splitting starting to occur. Because we remember that the cell, okay, it's these actual little clotheslines that are going to start to break apart from the center. From the center, they're going to start to actually retract like retractor beams. Okay, these centrioles are going to pull the clotheslines closer to them. And it's that force of that pull that actually cracks this centromere in two and is going to ensure that one sister chromatid goes in one direction and the other sister chromatid goes in the opposite direction. Okay? So we end up having one sister chromatid here from the X, one here, one here, one here, remembering that they are identical to one another. So in ensuring this, as these get pulled to opposite ends in anaphase, okay, we're ensuring completely equal DNA division. This is how it's accomplished. This is how both of these daughter cells are going to have the exact same DNA. Okay? So once DNA division, once chromosomal division has occurred, okay, some things start to happen here. We start to see something referred to as cleavage. Okay, that's actually where that word comes from. Okay, cleavage is basically a furrow found in rock. Well, the same thing starts to happen. We start to see this pinching in, okay, or the cleavage start to appear in this one larger cell. Okay, this is where we're going to start to make two cells. Okay, well, we also start to see that the nucleuses or the nuclei are starting to reappear, and we're left with our 4 little pieces of DNA, our little chromatids, in each of these. Okay? This is known as telophase. After telophase, and in your iBook and in your textbook, they refer to something called cytokinesis, which is actually the full-on division here of the cell. It's not actually a stage of mitosis. It happens after telophase to allow these tinier, okay, much smaller than the original parent, but to allow these actual bits of DNA here to uncoil themselves again, so that when this cell goes back into interphase, this actual interphase, okay, is actually different from the original cell's interphase because that original cell no longer exists. It's got two identical daughter cells that have the exact same genetic makeup that the mother cell had. So these two little guys here will in fact grow in size over time. And when it's their turn to divide, they will be the mother's size. They will be their own mother cell. Okay? Now, during this time, during interphase is actually divided into 3 chunks, right? We've got what's called G1, S, and G2. So G1, cell grows, okay? So the cell grows in size, right? S phase, the DNA, which right now is just a bunch of chromatids, is actually going to make a complete replica of itself because it's going to go from chromatid to chromosome. So that's where we go from just having one side of the chromosome or the X to making both sides of the X, right? That have the actual little centromere holding them in place, right? Each of these, if we remember from up top here, each of these is identical to one another, right? And then G2 phase, that is going to happen right before the cell's about to divide, and that is going to be rapid growth. And that is an everyday cell in your body's life cycle.